Greetings and welcome back. So, in the last vignette, what we did is we talked in fairly abstract and general terms about what I refer to as representations. And now, what we're going to do is we're going to dig a little bit more deeply into the mathematics of such representations. And what that's going to amount to is a generalization of the way that maybe people with physics backgrounds are used to thinking about vectors, although I would say probably most of us are already used to the sort of generalized notion that I'll be talking about here. So just as a reminder, I think, probably most people are used to the idea that a vector, like this one shown here, can be written as a linear combination of weights, which I'm calling x1 and x2, times the basis vectors e1 and e2. Those basis vectors are pointing along the two coordinate axes, as I show here. And some of you might be used to the idea that we expect that if we carry out a rotation of coordinates, then we can write the coordinates in the new coordinate system as a linear transformation of the old coordinate system, where we have orthogonal matrices, meaning that Q times Q transpose is equal to the identity. Another way of saying that is, just for our purposes, is that Q is a rotation matrix, and I leave it as an exercise for you to work it out here, but actually that was kind of hidden in the last vignette when we talked about the symmetries of the equilateral triangle. That was implicit. But what I want to do here is a different thing. So I want to start building up some ideas about vectors that are more abstract in some sense, and also they don't reflect coordinates in the conventional geometric three-space sense that I just showed you. So one of the examples that we're going to come back to a lot, and that I will use as a centerpiece of what I want to say about PCA and how to use data to try to figure out what the best coordinates are, and to try to ascribe meaning to some high-dimensional data, is that of coupled vibrations. So here at the top, I show you a coordinate system in which the positions of these two masses are the Cartesian coordinates x1 and x2. So x1 is how far the first mass is displaced from its equilibrium point. x2 is how far the second mass has been displaced from its equilibrium point. And I am free, if I want, so I'm interested in x1 of t and x2 of t, and I will find it very, very convenient to define a position vector or coordinate vector x, which is equal to x1 of t and x2 of t. And if I had three masses, it would be a three-dimensional vector. If I had four masses, it would be a four-dimensional vector, a column like that I've written here. So that's an example. But in the context of biology, we find ourselves often thinking of sequences, and those sequences themselves can be assembled as a vector. So here I show you the 142 amino acid letters that make up the one subunit of the hemoglobin protein. In the last vignette, I talked about how we represent ourselves, and so if we were to write a biography of me, or an autobiography of me, or a biography of you and an autobiography of you, it would be some sequence of letters. And the truth content of those letters we leave for others to try to work out, but I can represent all those letters as this very high-dimensional vector. We can also think of images in that way. So here what I'm showing you is a microscopy image of E. coli cells, two of them in particular. And what I want you to notice is that the intensities can be written basically as an array of numbers. So what I mean by that is, let's say that our image is 1,000 by 1,000 pixels. And so every point in that image has got an address. It's got an I point and a J point. So that's the Cartesian coordinates of that point. But I can write the entirety of that as one giant vector, where at each position in that vector, what I have is a number that represents the intensity. So I'm saying an image can be a vector. We're definitely going to get interested in the idea of cells and their mRNA counts. So I could have a vector. Let's see. I could have, I'm going to call it M of T. And this is number of copies of gene 1, number of copies of gene 2, and so on, dot, 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 all the way down to number of copies of gene M. So, up above I've written it as here as a row vector. Here I'm writing it as a column vector, but the key point is really that I can think of this object, which is the number of, I take some cell and I query it. I ask, how many copies do you have of the gene, the mRNA for gene 1, the mRNA for gene 2, and so on, and I align those objects in this very high dimensional vector, and then that raises the question, the very important question, which we will address, of how do we attribute meaning to that? Is there a way for us to reduce the dimensionality of that vector? Or is there a way for us to try to understand some linear combinations of that vector as being especially important, and so on? One of my favorite examples has to do with allele frequencies. So in this classic experiment done in the 1950s by Peter Bury, he was interested in what we now call genetic drift or neutral evolution. And the idea was that he prepared a set of flies, 8 males and 8 females, each of which was heterozygous, meaning that each one of them had one copy of a red allele, one copy of an orange allele, a white allele. And so the heterozygotes were as shown in the middle here. And so there were 107 vials. Each one of these vials has 8 males and 8 females. And what he would do is allow them to mate, collect the eggs, and randomly select 8 males and 8 females. And so what happens over time is that this population evolves even though there is no selection. The reason for the evolution is essentially the law of small numbers, which is to say that if I were to ask 4 people whether they were born on an even day, many times I would find not 2 evens and 2 odds, but I'd find 3 evens and 1 odd, etc. And so here the partitioning is random, and because of the small numbers, sometimes there's an excess of red, sometimes there's an excess of white. And the reason that this comes up in the context of this discussion of vectors is that the allele frequencies, so let's call this the frequency at generation n, is going to be 1 over the number of flies. And then this first element is number of flies with 0 red alleles. And then the next entry is number of flies with 1 red allele. And we go all the way down until we get number of flies with 32 red alleles. Why do I choose these numbers? Because I told you that there's 8 males and 8 females for a total of 16 flies. That means there's a total of 32 copies of our allele of interest. This vector is 33-dimensional. Okay, so the point is that, and note I divided by 1 over n flies. So this is a frequency vector, and the reason for n here is this is then the nth generation. So in other words, I'm keeping track of the number of red alleles in each generation, and in the nth generation I can write that as a frequency vector. It's a 33-dimensional vector, and each entry in that vector tells me the frequency in the population of a vials that contain a certain number of copies of the red alleles. Okay, so what was the point of this whole brief vignette? The point of it was to encourage us, to get us used to the idea of a generalized notion of vector beyond the simple geometric 3-space vector r is xi plus yj plus zk, or as I will write it in this course, x1e1 plus x2e2 plus x3e3. So we've given several examples, and what we're going to do now is we're going to try to actually work out the mathematics of how to capture these types of vectors, and then given those vectors, to try to ascribe meaning to them by finding the best coordinate system. And it will become clear what I mean by that as we proceed.